Okay, so this is what we are going to do today. Uh, yes, of course, our BDD syllabus is over, right? Our BDD, at least theory syllabus, is over. So I have given you the practicals for you to try those. Okay. So since we have our BDD slot available today, I thought of using that for completing out this BSO topic. Okay. So today afternoon, instead of BDD, we would be, conducting the practical of BSO. I'm going to teach you two topics from BSO, right? Biomolecular structure and organization. Okay, so the very first topic that I am going to deal with is the practical aspects of protein carbohydrate interactions. Okay, and the next topic that I'm supposed to teach you, which we will continue tomorrow. Tomorrow we will be talking about RNA structures. Okay, तो कल अपन RNA structures के बारे में बात करेंगे और आज अपन प्रोटीन कार्बोहाइड्रेट इंटरेक्शन के बारे में बात करेंगे. Is it clear? So this is what I am going to engage you for today and tomorrow. Okay. So I hope कि Dr. Vijay has completed your protein interaction. हुआ क्या? नहीं हुआ है?
No, sir, only carbohydrates
No,
he has
sir.
taught us.
Okay, okay, हो जाएगा. Don't bother with that. Okay, so I will first begin with protein carbohydrate interaction. Okay, इसके साथ में और दो टॉपिक्स है, like protein nucleic acid interaction और protein interaction as well. So I will be taking care of this protein carbohydrate interaction. Okay, the structural aspect of protein carbohydrate interaction. Okay, so now when any carbohydrate moiety goes and attaches to any protein. Okay, so you can say that there are three major classes of interactions that will stabilize such protein and carbohydrate interaction. Okay, so now I hope you know or you are aware of what are carbohydrates, right? तो यहाँ पर we are going to talk about the structural aspects of how these carbohydrate interact with the protein molecule. Okay, and when such interactions happen and when such protein carbohydrate interactions or when such protein carbohydrate complexes are formed, these protein carbohydrate complexes are stabilized by three major type of biochemical interactions. So first category you can consider them as polar interaction, and polar interactions are usually in form of hydrogen bonds. Okay, so now I will when we begin this thing, I will let you know what is the role of hydrogen bonding in protein carbohydrate interaction. Okay, we will also try to classify these different type of hydrogen bonds that occur in sugar interactome. What I mean to say over here is that how can we classify different type of protein carbohydrate hydrogen bonding as such. Okay, in which I will introduce you with the concept of what is cooperative hydrogen bonds, what is bidentate hydrogen bonds, and what is network of hydrogen bonds. Okay, so this hydrogen bonding would be one of the major focal point of interest while we analyze the protein carbohydrate interaction in more details. Okay, well, beside this polar interaction, these carbohydrate also interact with your protein molecule using apolar interaction, like it might be stabilized by hydrophobic it can be stabilized by van der Waals contact. Okay, it is said that it can also form pi electron clouds based interaction, but I really doubt about formation of pi electron cloud or pi stacking interaction in protein carbohydrate complexes. I will go in more details of the speculation about why pi stacking interaction is not feasible, but in most of the paper, they show ki pi stacking interaction happens with the carbohydrate moiety with say some residues of protein molecule. Well, we will talk about that in a little while, right? So during practical, we would talk about in more details of it. Then there are a few interactions that are known to stabilize such protein and carbohydrate complexes, which can include salt bridges, that is ionic, and some of the carbohydrate moiety can also interact with ions, which in turn interact with say some residues of proteins as such. Okay, so that is called as coordination with ions. So say for brief, we won't go in details of this coordination with ions. So basically this is supposed to be your practical session, right? But unless and until you are aware of say some theoretical background, directly going on to practical and showing you how do we visualize the protein carbohydrate interaction would make very less sense. So that is my objective first, to get you a brief idea about what we are going to deal with in our next practical session after lunch. Okay, so this is what my objective is. So today, whatever time we have got prior to lunch, I would briefly explain what are different type of interaction that happens in protein carbohydrate complexes. Okay. ठीक है. So well, before we move on, one might ask a question कि भाई, why we are interested in learning this protein carbohydrate interaction? What might interest us? क्या फायदा होगा ये प्रोटीन कार्बोहाइड्रेट इंटरेक्शन या फिर प्रोटीन कार्बोहाइड्रेट कॉम्प्लेक्स का स्ट्रक्चरल एनालिसिस करके? ठीक है. So the thing is that there are therapeutic application of such protein carbohydrate interaction. One of the example I'm going to explain you here, that is already published in this particular paper back in 2011. You can download this paper and go into more details of how protein carbohydrate interactions is being used in therapeutic context as such. Okay, so now just to begin, that you must be aware of influenza virus. Okay, so this influenza virus, when it starts to bud from the host cell, okay, so it is said that the mature viron is connected connected to the host cell by these neuraminidase. Okay, बात समझ में आ रही है आपको कि this is receptor, it has got the host cell has got certain receptor that contains sialic acid. Okay, and to this sialic acid receptor, your budding viron is somehow connected. Okay, so now this virus expresses an enzyme called as a neuraminidase, and this neuraminidase is responsible for cutting off the sialic acid bond that connects the viron to this receptor. Okay, and hence after the activity of neuraminidase, this bond with sialic acid that connects the viron with the receptor, okay, that particular bond is broken and this mature viron is thus released. Okay, so now therapeutically what happens is that people have created such neuraminidase inhibitor like Tamiflu, what we call it as oseltamivir, or there are another neuraminidase inhibitor like zanamivir. Okay, so what do these neuraminidase inhibitor do is that they go and attach to the neuraminidase and hence inhibit its activity. Since neuraminidase remain in inactivated state, there is no one to cleave the bond between sialic acid and the viron particle, so that this neuraminidase inhibition would prevent the escape of the mature viron from the host cell as such, and hence viral infection is halted. So now this sialic acid is, one of the carbohydrate based moieties. Okay, so this is what it happens. Host cell has got sialic acid by which it is connected to the viron particle. Okay, so drugs like oseltamivir and zanamivir goes and attaches, and hence it prevents the viral uh, replication or viral escape from the host cell as such. Okay, so now this oseltamivir, which is a which is, you can say, an analog of sialic acid, and this sialic acid is one of the carbohydrate based moiety. ओके, दिख रहा है यहाँ पर आपको, इट इज वन ऑफ द कार्बोहाइड्रेट बेस्ड मोइटी. तो ये साइलिक एसिड का ही एनालॉग है आपका ड्रग ओसेल्टामिविर, which somehow goes and attaches to the active site of oseltamivir or Tamiflu, what we call it, and it since it is occupied by this Tamiflu, this neuraminidase is unable to catalyze the breakage of the sialic acid residue and host cell bond, right? And hence the viron remains indefinitely attached to the host cell, and hence they cannot escape from the host cell as such. Okay, they cannot detach themselves from the host cell, and hence the virus life cycle is halted. This is, now you get key how important is protein carbohydrate interaction. If we don't know the protein carbohydrate interaction in more details, we would never be in a position to generate or to synthesize drugs like Tamiflu, which eventually helps in controlling the diseases like H1N1, influenza A H1N1, okay, or influenza A flus. Okay, so this is one of the application. Okay, well now let us talk about what are the forces that stabilize such protein carbohydrate interaction. Okay, so one of the main forces that are known to stabilize such protein carbohydrate complexes are in form of polar interaction, and most common polar interaction that happens in biological world so far your proteins are concerned are hydrogen bonds. Okay, so there are three major roles of hydrogen bond plays three major roles in protein carbohydrate interaction. First is that they provide stability to the protein carbohydrate complex. So the hydrogen bonds are stable enough to significantly contribute the ligand's binding affinity. Okay, they are also known to confer specificity. Okay, so van der Waals forces are non-directional in nature. On the other hand, your hydrogen bonds are highly directional. So these being directional, it is said that this hydrogen bonding might confer the specificity for protein carbohydrate interaction. And of course, these hydrogen bonds are sufficiently low strength to allow rapid ligand dissociation, and therefore they are helpful in controlling the dynamics of protein carbohydrate interactions. Right? They control dynamics. How frequently or how frequently your protein molecule would go and form complex with the carbohydrate molecule. Such questions can be answered through the control dynamics that is eventually controlled by formation and breaking of various hydrogen bonds between the protein carbohydrate complexes. Okay, so now the protein side chains at the sugar binding site are usually in form of aspargine, okay, ASN, aspartate, glutamate, glutamine, arginine, and histidine. Okay, so there are planar residues that are fixed, or they have very little freedom of torsion rotation. Okay, so due to this, interaction with such residues brings more stability to your protein carbohydrate complex, and in this way your protein carbohydrate complexes are very, very stable. Okay, and this happens due to protein, you can say hydrogen bonding, that happen in protein carbohydrate interaction. Okay, so now you people might be aware that this carbohydrate moieties, well, they have got hydroxyl groups, okay, in their structure, and this hydroxyl group eventually plays very important role in formation of such protein carbohydrate interaction. Okay, so this hydroxyl group, well, the hybridization state of this oxygen in such hydroxyl group of carbohydrate moieties, it is, the hybridization states are arranged in such a way, the orbitals are arranged in such a way that it can act as an acceptor of two hydrogen bond, and due to the presence of lone pair of electron on the oxygen, it can also act as donor. Okay, so this hydroxyl group of carbohydrate are the one that would participate heavily in formation of hydrogen bonds. Okay, moreover, these hydroxyl group are stereospecific and they are highly exposed. Therefore, they can effectively form hydrogen bond with the amino acid residue in the carbohydrate binding region of the protein molecules as such. Okay, so now, since I have already told that this oxygen is in sp3 hybridized state, and hence it contains a lone pair of electron. Okay, and due to presence of this lone pair of electron and hydrogen that is attached, okay, it can act as a donor of one hydrogen bond, okay, due to the lone pair of electron, and it can act as an acceptor of two hydrogen bond from, well, say for example, the carbohydrate moiety as such, or protein moiety as such. Okay, so due to specific hybridization state of this oxygen, it enables the carbohydrate moieties to form three hydrogen bonds at a time. Okay, and this is going to be a very interesting fact in our further course of practical, जो मैं आपको लंच के बाद में बताऊंगा. But for time being, just remember that due to the sp3 hybridized state of oxygen, the hydroxyl group in your carbohydrate can form three hydrogen bond, in which it can act as a donor of one as well as acceptor of two. Okay, सबको ये बात समझ में आ रही? Is it clear?
Yes, sir.
Yes, sir.
Okay,
Yes,
please
sir.
let me know if I am going fast. Okay, so we will come to this thing when we do our practical, right? Okay, now let us talk about cooperative hydrogen bonds in sugar. Okay, so these cooperative hydrogen bonds result from simultaneous participation of sugar hydroxyl as a donor and as an acceptor of hydrogen bond, as I have already told you. Okay, तो ये डोनर और एक्सेप्टर दोनों कैसे क्या हो सकता है? ड्यू टू sp3 हाइब्रिडाइज स्टेट ऑफ ऑक्सीजन, जैसे कि मैंने यहाँ पर बताया था. Okay, since it contains a lone pair of electron, it can act as a donor. Okay, it can act as acceptor. Okay, one oxygen act as acceptor and more than one NH can act as donor. अभी यहाँ पर देखो, focus on this region of protein carbohydrate interaction. इसमें क्या दिख रहा है आपको? two amino group are forming hydrogen bond with this OH group. Okay, and that is possible. Why? Because it contains a lone pair of electron which can act as an acceptor of these two hydrogen bonds. Okay, and it has got one OH group and it has got hydrogen group over here which is attached to oxygen, which can act as a donor to form hydrogen bond with glutamate 14. And this particular interaction is termed as cooperative hydrogen bond. बात समझ में आ रही है आपको? ये लोन पेयर ऑफ इलेक्ट्रॉन रहने की वजह से it can act as an acceptor of two hydrogen bonds, and these two hydrogen bonds are donated by whom? ASN 232 and another from ASN 205. Okay, and since it contains a hydrogen, it can also act as a donor. In such a case, it acts as a donor when it forms hydrogen bond with this glutamate 14 as such. So such intricate network of hydrogen bonds are termed as cooperative hydrogen bonds. Okay, so now not all of the carbohydrate moieties can form, or you can say not all hydroxyl group are probable of forming such cooperative bonds. Okay, in case of glucose, all hydroxyl groups are equally probable for forming such cooperative hydrogen bonds. On the other hand, in case of carbohydrates like arabinos, only the hydroxyl groups that are placed on position 2, 3, and 4 are able to form such cooperative hydrogen bonds, others cannot. Okay, so this is what I want to make clear. Okay, then other type of hydrogen bonds that you can see is in form of bidentate hydrogen bonds. तो देखो एक और डिफरेंस मैं यहाँ पर बता देता हूँ कि कोऑपरेटिव हाइड्रोजन बॉन्ड जब तैयार होता है, then each and every hydrogen bond is donated by different amino acid residue. This first hydrogen bond is donated by ASN 232. This second hydrogen bond in your cooperative hydrogen bonding is donated by ASN 205. And the acceptor of hydrogen bond is from glutamate 14. दिख रहा है यहाँ पर? Is from glutamate 14. All in all, what I want to say is that when cooperative hydrogen bonding is concerned, different amino acid residues are involved in formation of hydrogen bonds with the hydroxyl group of sugar moiety. Okay. Now, in contrast to this, when we will discuss this bidentate hydrogen bonds now, the bidentate hydrogen bonds are contributed by a single amino acid residue. Do you see over here? This bidentate hydrogen bonding is contributed by single amino acid residue ASN 232, wherein two hydroxyl groups are engaged in hydrogen bond with two atoms from the same amino acid residue. That is ASN 232. That was not the case when cooperative hydrogen bonding happened. In cooperative hydrogen bonding, multiple residues were involved in formation of those three hydrogen bonds. While in case of bidentate hydrogen bonding, a single residue forms two hydrogen bonds, but with two hydroxyl groups of sugar moiety. In contrast, in cooperative hydrogen bond, a single hydroxyl group is involved that is forming hydrogen bond with multiple amino acid residue from carbohydrate. Sorry, multiple side chains from protein molecules. बात समझ में आ रहा है आपको? What is the difference between cooperative hydrogen bonds and bidentate hydrogen bonds? Do you want me to repeat it?
Yes, sir.
Yes, sir.
Okay. in cooperative hydrogen bonding, what happens is that there are different amino acid residues that forms 3 hydrogen bonds with a single hydroxyl group. यहाँ तक clear है?
Yes, sir.
On the other hand, when we are talking about bidentate hydrogen bond, single amino acid residue will donate 2 hydrogen bonds. यहाँ तक clear है? Is this fact clear?
Yes, sir.
Yes, sir.
Yes, sir.
अगली difference क्या है कि when bidentate hydrogen bonds are formed, it involves 2 hydroxyl group from the sugar. दिख रहा है यहाँ पर?
Yes, sir.
On the other hand,
Yes,
when
sir.
we are talking about cooperative hydrogen bond, single hydroxyl group from the sugar is involved in formation of multiple hydrogen bonds. Right? So this is the basic difference between cooperative hydrogen bonds and bidentate hydrogen bonds. It differs in terms of how many amino acid residues are involved, and it also differs in terms of how many hydroxyl group from sugars are involved in formation of hydrogen bonds. Okay?
Excuse me, sir. Sir, I had to ask one thing regarding the cooperative hydrogen bonding.
Yes.
Sir, the number of the hydrogen bonding will be same as that of the number of the protein, means the amino acid residues, because each amino acid is going to.
For a separate hydrogen bond.
Yes, sir. So,
Yes.
is the number going to be same?
Number in the sense I don't get your question.
Sir, I'm saying that the number of the hydrogen bonds that will be formed in any protein structure is going to be same as that of the number of as that to the amino acid residues.
Okay, let me make one more thing clear. Right? Cooperative hydrogen bond में 3 hydrogen bond होते हैं.
Yes, sir.
Bidentate hydrogen bond में 2 hydrogen bonds होते हैं.
Yes, sir.
Okay. कोऑपरेटिव हाइड्रोजन बॉन्ड में जो कुछ भी 3 हाइड्रोजन बॉन्ड्स तैयार होते हैं वो अलग अलग 3 अमीनो एसिड रेसिड्यू से आएंगे.
Yes, sir.
तो जहां जहां पर भी कोऑपरेटिव हाइड्रोजन बॉन्डिंग होगा आपके कार्बोहाइड्रेट और प्रोटीन मॉलिक्यूल के बीच में, तो हर एक कोऑपरेटिव हाइड्रोजन बॉन्डिंग पैटर्न में 3 अमीनो एसिड रेसिड्यू रहेंगे.
Okay, sir.
Okay.
Yes, sir.
Is it clear?
Yes, sir.
Okay. Over here situation is different. यहाँ पर हाइड्रोक्सिल ग्रुप कितने रहेंगे? In case of bidentate, bidentate हाइड्रोजन बॉन्ड में हाइड्रोक्सिल ग्रुप कितने रहेंगे?
2.
2.
और अमीनो एसिड रेसिड्यू कितने रहेंगे?
1.
1.
हाइड्रोजन बॉन्ड्स कितने होंगे?
2.
2.
शाबाश, आगे बढ़ सकते हैं अभी. ओके, थिंग्स आर क्लियर नाउ. ठीक है. नाउ लेट अस टॉक अबाउट नेटवर्क ऑफ हाइड्रोजन बॉन्ड. ठीक है, तो बाइडेंटेड हाइड्रोजन बॉन्ड्स एज वेल एज कोऑपरेटिव हाइड्रोजन बॉन्ड्स के अलावा there is also possibility that the water molecule that are present in vicinity of your carbohydrate moiety can also engage themselves for bridging the hydrogen bonds that might happen between the carbohydrate hydroxyl and the amino acid residues side chain. Okay, say for example ये वाला हाइड्रोजन सॉरी ये वाला आप H2O consider कर सकते हैं. Well, this water molecule with number 309, okay, so this water molecule is supposed to bridge this NH2 group के साथ में एक हाइड्रोजन बॉन्ड हो रहा है with that of glutamate 4. दिख रहा है यहाँ पर? ये वाटर मॉलिक्यूल कौन सा हाइड्रोजन बॉन्ड ब्रिज कर रहा है? The one that comes from NH2 of ASN 205 to the glutamate 14. दिख रहा है? Okay. While this one, ये जो water molecule है at 310 position, it is bridging your carbohydrate moiety to form a hydrogen bond with this aspartate. Can you observe this thing? Right? So ये जो intricate network है that arise due to presence of water molecule which bridges different hydrogen bonds along with the cooperative hydrogen bonds, bidentate hydrogen bonds and normal hydrogen bonds, and collectively it is called as networks of hydrogen bond.
Excuse me, sir.
हाँ बोलो.
Sir, can you please explain bridging of hydrogen water molecule 309?
कहाँ पर गया 309? This, right?
Yes, sir.
So देखो ये water molecule कितने hydrogen bonds को bridge कर रहा है? The one that is forming between ASN and glutamate, right?
Yes, sir.
ASN का जो नाइट्रोजन दिख रहा है that is forming hydrogen bond with H2O. This H2O which in turn is forming hydrogen bond with glutamate 14. Okay, it is simultaneously bridging the hydroxyl group with that of glutamate 14. दिख रहा है यहाँ पर hydroxyl group यहाँ से it is forming hydrogen bond with H2O at 309th position, which in turn is bridged with hydrogen bond with glutamate 14, right?
Yes, sir.
So in this way, water mediated complex or complicated network of hydrogen bonds that is formed along with your bidentate normal hydrogen bond as well as cooperative hydrogen bond, those are called as, you know, network of hydrogen bonds.
Sir, this bridging could happen also in cooperative and bidentate also.
Yes, if it is involved with water.
Okay.
Okay.
Okay, sir.
Okay. So now let us talk about this apolar interaction in form of hydrophobic interaction. Okay. So this hydrophobic interaction is a characteristic steric deposition of hydroxyl group that creates hydrophobic patches on sugar. Okay. तो क्या हो सकता है कि ये जो hydroxyl group है, they can be oriented in a specific way that would expose only the carbon portion of your carbohydrate moiety towards one end. Okay, and in such a way it can create a hydrophobic patch on sugar surface, and such hydrophobic patch on sugar surface can now interact with the hydrophobic amino acid residues from the protein. So what hydrophobic amino acid residues are present in your protein? They are in form of valine, leucine, isoleucine, right? बात समझ में आ रही? तो हाइड्रोफोबिक पैच आपके कार्बोहाइड्रेट पर कैसे क्या तैयार हो सकता है? When the hydroxyl groups in your sugar moiety get, you know, oriented in a specific way, that would lead to exposing only the carbon region of your carbohydrate in one end, and that would result in formation of a hydrophobic patch on the surface of your carbohydrate moiety. And such hydrophobic patches can be now be stabilized by hydrophobic interaction that involves residue like valine, leucine, isoleucine from your protein molecule. Okay, ये समझ में आ गया? One might ask ना कि sir, now our carbohydrate moiety, since it contains OH group, how hydrophobic interaction can happen over there? बरोबर है? तो उसका रीजन क्या है कि this hydrophobic interaction or hydrophobic patches can be formed on hydrogen bonds when hydroxyl group of your carbohydrate would orient themselves in a specific direction. that would expose all the carbon atoms from your carbohydrate moiety on one of the end of your sugar molecule as such. And that can create a hydrophobic patch, which can be stabilized by hydrophobic amino acid residues like valine, leucine, and isoleucine. Okay. So now this is what I am talking about. तो देखो ये जो पेपर है डेल कार्मेन फर्निएंडेज also that was published in 2012, current protein peptide science. So I have taken figure from this thing. Okay, so what these guys are saying that these carbohydrate moiety can form pi stacking interaction with this W. W यानी कौन सा अमीनो एसिड रेसिड्यू होता है? टायरोसिन या ट्रिप्टोफैन?
Tryptophan.
Well, now I don't understand such logic. So देखो, in order to form pi stacking interaction, so pi stacking interaction are stabilized by pi electron clouds. Okay, so I don't have any issues on the fact that tryptophan has got its own ring that can have, that can have its own pi electron cloud. But the carbohydrate moiety में के जो carbons होते हैं, well, they are in sp3 hybridization state, right? And since they are in sp3 hybridization state, they won't have any pi electron cloud over there. Okay, तो pi electron cloud कब तैयार होगा आपके cyclic ring में? When all of the atoms in your cyclic ring are in sp2 hybridized state. तो अगर sp2 hybridized state में रहेंगे जब भी आपका रिंग प्लेनर रहेगा. Okay, and unless and until the ring is planar, pi electron clouds cannot form, right? But still there are some paper that discuss कि it can form pi stacking interaction, right? So there are categories of pi stacking interaction. So what I can at most agree with is that this protein carbohydrate interaction at most can form pi alkyl interaction. That is, the pi electron cloud from one of the residues like phenylalanine, tyrosine, histidine, and tryptophan can somehow form interaction with the electron cloud from alkyl groups. ज्यादा से ज्यादा मैं यहां तक अग्री हो सकता हूँ ये पेपर के साथ में. Okay, so this is my agreement. This is my argument whether it can form pi stacking interaction or pi alkyl interaction. Okay, तो मे बी यहाँ पर जो बताया गया है that is pi alkyl interaction. Okay, तो pi stacking के भी types होते हैं. It can be pi stacking interaction, it can be pi alkyl interaction, it can be pi T-shape interaction. Okay, there are different types. You can, if you have installed Discovery Studio, you can visualize this pi alkyl interaction, pi T-shape interaction in more details. They will let you know what are the residues that can participate in such pi alkyl, pi stacking, or pi T-shape interaction. Okay, well, let us not go in more details of this apolar interaction that involves stability by interaction of pi electron clouds. Okay, so this is what it is. So in our practical session, after after your lunch at 2:30, we would begin with visualizing all of these protein carbohydrate interactions. Okay, तो theoretical portion आपकी समझ में आ गया?
Yes, sir.
What is
Yes, sir.
pi interaction, and what are different type of hydrogen bonds, and what are apolar interactions that are involved. Okay, so if that is the case, I am done with it. Okay, so we would be dealing with the practical aspect in post lunch session. So see you at 2:30 then. Okay, is it
Excuse
clear?
me, sir.
हाँ बोलो बेटा.
Sir, actually, I was facing network issue during attendance time.
Okay, what is your name?
Gaurav Kumar Singh, 20 roll.
Okay, बेटा ठीक है, कोई दिक्कत नहीं. I have added P over there. Okay, so now maybe I will send you a Zoom link if there is nothing, or maybe I will send you a Google Meet link if Zoom is occupied. Okay, ठीक है. So whatever it is, I will send a link on your class group, right?
So can we have it on Teams, if not Google, because we are not able to record it there.
No, I will record it on my end over here if we go it on Google Meet. You don't worry about that. All you want is a recorded lecture, right? Okay, I will give you that. You don't worry about it, right?
Thank you, sir.
Okay.
Yes.
Okay, now I am stopping the recording. Huh?